I see.
Green button.
Okay.
No?
So I'm going to use some slides just from Aviv's previous conversation. And yeah, this is a great time. I just came from the previous meeting. I've been here for a few days from the single-cell genomics meeting, and the discoveries are amazing. We've discovered new disease-meaningful cell types. This is from Aviv and Jay right here. New analysis methods, new techniques. So it's really exciting, and we feel good that we're atlasing. I've even heard people call this an atlas, but I call this a cloud. And this meeting is about going from a cloud to an atlas, to something that is comprehensive and complete and spatial at both the histological and the organ level, and that you can query and get statistics and concrete, rigorous scientific answers. And in order to do that, yes, we need to collect and assemble. And the first thing we need to design is what we're going to collect and how we're going to collect so that we can assemble it all back. And in order to do that, I think you should take one step back and ask, what is the biological questions? And I've sat in many of these breakouts, and we can think of a million things, because what do we want to do at the end? We want everything, because we want it comprehensive, but we also need to feel good along the way. We need milestones. So I want everyone in the breakout to take a little thought experiment. If I gave you $10 million, what is the key biological question you want to address most? What is the most important biological questions of your fields that such an atlas can address? And what data do you need to collect so that it will be coherently and with statistical power and scientific rigor give you what you want?
Excellent. So this is, again, following on from what Aviv talked about. There are absolutely key challenges in experimental design. You can only do so much with statistics. If the experiment is designed badly, you just cannot correct for terrible batch effects. You can't correct for data that is not compatible. So we need to think very seriously, and this is something to do in the breakout sessions about experimental design. So in the context of what Aviv mentioned in terms of the spatial landscape, and this is work together with Aviv, Raul, and Alex Richko, we've got to think about what are the features that are going to define a particular tissue. So if you've got multiple, say, samples from the same organ, what are the key features that are going to allow you to say, well, I've got this sample relative to that part of the organ? So are there key landmarks? What are the anchors that we're going to use to combine different data? In something like the brain, it's relatively straightforward because it's got a very highly structured setup. But other organs, it's going to be much more challenging. So we need you to think, what's special? What's important about your system? What are the features that we're going to need to build a comprehensive and rigorous atlas? Dana mentioned this already, but it's really important to reiterate. What are the biological questions that your domain would most like to answer? How are you going to make sure that your data are usable? And all of this is going to dictate how the data are analyzed. But as mentioned, one can't do anything if the data aren't generated appropriately. If an inappropriate experimental design is used, it's simply not possible to do anything sensible. A key thing from an analysis working group perspective, we don't want to analyze the data from each pilot. This isn't about us accruing data and doing all of the analysis here. But we do want to help guide the development of computational tools that are going to facilitate creation of a comprehensive and useful atlas. And in order to do this, we need to interact perhaps more as a community from the different atlas projects that are focused on tissue with the analysis working groups so that we can work out what the important questions are and how we're going to try and build this. And Peter, at the end of this, is going to give a really nice example of one application of a tool that we think has potentially high utility across the whole of the human cell atlas. Okay, so in order to stimulate these discussions, we've got to think, what are the important use cases? What could go wrong if I don't design the experiment well? So if you're comparing, say, diseased and normal, we need to have a comprehensive enough normal atlas that if we see something different in a disease sample, we know that statistically that is actually genuinely different. So we just need to think about these sorts of things. We need to think about the sort of metrics for comparing normal and disease. We need to think about the coordinate framework. And critically, and this is really encouraging as a community, because this community is, in some sense, this atlas is driven by two computational biologists, but it's important to say that it will thrive when biology and statistics properly interact with one another. This is really key. And we can only do this as an analysis working group if we have rigorous engagement and comprehensive engagement with each of the projects. So just a bit more brass tacks, the current state of play. The analysis working group is composed of 8 members, and there's 3 active subgroups. There's one, and oops, I don't want to emphasize this too much. Go back. Yep, on that works around the data coordination platform, in particular interacts with the analysis portion, so led by Tim Tickle, and the DCP, and you'll hear more about this later. There's also a group that's devoted to thinking about new methods for experimental design. And then we have another group that's thinking more about the interpretation and how we can make these data really accessible to the wider scientific community, so focusing more on the portal aspect. In terms of other things we've done more concretely, we've organized 2 Jamborees. This is a picture from the last one, held actually within this institute earlier in the year. And this Jamboree is really important to us, and we're really grateful to the support from CZI for enabling this, because it allows us to get the whole community, postdocs and students, really the people who are actually doing the work, not just designing the experiments, but actually doing the work, to come together and to focus on really key problems. And it's very consistent with what we've spoken about today. This last Jamboree focused on challenges in experimental design, and Dana, over the next few slides, is going to talk about exactly what they came up with.
Yeah, I want to again stress that it's really wonderful to work with the young people, to bring young people all together. Important component of it, again, is building community and mixing people across labs. The AWG set up some specific tasks, and part of the Jamboree is actually designing a specific and concrete task. And I'll tell you one of them, just to give you a flavor of what this thing looks like, so it won't be too airy. There's this idea where we do a skydive to be as comprehensive as possible, to get all the cell types, to be unbiased. But then, because there are big differences, sometimes 5 orders of magnitudes, or 6, between rare and non-rare cell types, we have to dive deeply. We have to drill down and sort or enrich through some technology for those cell types that we miss. And how do we do that? How do we build the statistics, the algorithms, and the methods to actually deep dive? So we made it concrete, and we got a data set that was generated in Aviv and Arik's group that they gave us right after generation, even before they analyzed it, 350,000 plus cells. Ambrose Carr, who was already out of my lab, sat for weeks and developed an in silico setup that allows people to query this data, to do sort experiments, and to really measure distances and metrics, all in this beautiful Python Notebooks setup. And they got a concrete task. You get 10,000 cells, and you have 6 sorts with 6 markers for 10 more thousand cells. How close are you going to be? And what really blew my mind away is what happens when you bring these young people. It made me feel like maybe I should retire, because they broke all boundaries and all boxes. And we were thinking of these boring gates, and they said, why boring gates? Let's hack the fax machine. Let's actually build an arbitrary function of these 6 markers. And let's be probabilistic, because we don't know. There might be regions we don't know to look at. So let's not narrow down on one little region, but let's actually understand more fundamentally, where are we missing cells? Where are we not getting the cell state and the density well? And enrich for stuff, just basically build a negative gate in 3 different groups for each sort of the same solution, and build a new statistical metrics for where are we sampled well and when have we not sampled well. New computational and mathematical methods to evaluate where we should sample, to actually choose markers that allow us to evaluate this. Probability distributions, they invented a new field. 50 people, 2 days, no sleep, brilliant minds, good atmosphere, just invented an entire new field. And lots of food, yeah. And they changed the way we think about things. Density isn't the right thing. It's how well we've sampled the density. And these 2 plots are actually from their final slide. We gave them an hour to come together, and they, in an hour, merged the ideas of 3 different groups, invented new math in that hour, and showed how they can sample and capture the diversity of the system much better with the new ideas that they built. And now the experimental design group is trying to follow up on some of these things and make them more concrete, so that we can provide you tools to evaluate what parts of the atlas, what parts of the tissue, what cell types and cell states have you analyzed well, and where might you have more to investigate. When you have a tissue that's very similar to what's been found, and when are you onto something new? And the experimental design working group is following up on these things. And finally, the portals group, we're going to give you another concrete example of something that's been thinking of. This is really spearheaded by Peter and Fabian. So something that we hope could help you, and we would love your feedback on it.
Yeah, so this is an example of a tool that we think might help to leverage the expertise within the community and encourage people to share their expertise. This is just a snapshot of the tabula muris, sort of mouse atlas. You know, we understand that atlas is going to be pretty big. And of course, the data, even if you have tons of data, it's only as good as your ability to interpret it, right? And so a key, I think, a secret that doesn't come through in the papers and all the successes that we have is that the interpretation process is actually quite painful and very much manual. You pretty much have to, well, first of all, representing the data, and that's probably a statistical part. It can be a trick, but also once you've done all the statistical analysis, you need to be a tissue expert. You need to understand what's going on with this particular experiment in order to actually assign, let's say, even plain labels on the subpopulations that you're observing. And so this manual process obviously doesn't scale very well and doesn't transfer very well in terms of knowledge between different groups. And so ideally, you know, we would like to basically have automated tools to do this, or at least guess, you know, what the labels are, what the processes that are taking place within the data. And you know, I think as far as machine learning is concerned, there are plenty of opportunities here and plenty of expertise. However, we very much clearly lack the knowledge or database that, you know, that would give us the basis for training such tools. And if we actually want these machine learning tools to be advanced, to handle different contexts, to handle, say, you know, normal disease states, to handle sort of a variety of processes, not just kind of top-level labels about, you know, CD4, CD8 T cells, we want probably a variety of opinions about individual processes that are being observed, individual subpopulations that are being observed, so that machine learning tools can actually build on this and try to give a rich output. So what we're basically thinking about here, proposing kind of a centralized annotation system, where we would have basically a cloud-based database that would keep track of annotations that people have made on their data sets or on shared data sets, or on anything else that they're willing to sort of share with this system. And the basic purpose of this is to allow to deposit annotations and to read annotations with respect to any particular data set, and provide also basis for machine learning tools that would be able to eventually start automating this process. And again, I don't think it will happen on the day one, but as we accumulate this data, it should get better and better. And so I think the effort we're thinking about is to provide sort of this infrastructure, particularly the APIs, sort of the communication channels for all these boxes to talk to each other, and provide some reference implementations as far as annotation clients are concerned and some of these propagation engines. So I'll give you an example just so it doesn't look abstract. So there are existing data exploration tools, like Cell by Genes effort, for instance, is building a UI. We have one in our own lab, also Loomis, CMAP, and Spring, that allow you to browse the data sets and look at different subpopulations. And so we can extend some of these tools to provide sort of reference implementation support so they can talk to the database, right, to the centralized annotation database. And so if you're using one of those tools then, and you have a new data set, for instance, or let's say this data set has been shared before, and you're trying to look for, you know, whether there are existing annotations for this, it would give you a listing. So we want more than one opinion, to be honest. And some opinions will be, you know, more accurate than others. And here, my opinion doesn't score very well, but it's okay, you know, as long as we have a system of prioritizing it. Somehow, we actually want, especially for these machine learning tools, to be able to have a variety of opinions. And once you select it, basically, it will just give you, let's say, clusters in this case, and you can go on and analyze it. We also want to keep track of explanations. You know, what evidence did that person, an expert, see in order to actually make that label, and how to capture that properly is going to be an issue. Now, the other function is that if you actually consider yourself an expert, you can try to deposit annotations, make new annotations, and you know, a lot of these tools would allow you to select, let's say, a subpopulation, and then we would ask you for labeling. Now, we do want to make this somewhat structured. I mean, ideally, everybody would play by the rules and use an ontology, but I don't think it's realistic. We do want to have a low barrier of entry so people are not afraid of using it. So I think we will try to work with HCA, for instance, teams that are working on ontology to make sure we have suggestions for ontology, standardized terms, and and maybe more specific terms if they're not being very specific in the annotation, to encourage people to use standardized terms and go deeper. Same thing with evidence. We like structured evidence, but we understand we won't always get them. So you can annotate other things, for instance, gradients of processes. The automated annotations is where we would like to get to. Again, it's not going to happen on day one. You might have this annotate my data button, but it's going to be very primitive. And eventually, we want to have a choice of engines. That's why we have these boxes with APIs and so on, so you can select an engine that would actually do an annotation. And somehow, we don't do well here as well. But granted that you have that choice, and we have sort of standardized ways of exploring and finding these engines through these APIs, different tools, exploration tools, should be able to take advantage of this. And once you run this, it will give you some sort of an annotation. All right, so that's the overall proposal, and I think the key thing here, we're not doing it just yet, right? We're thinking that this could be a useful thing for the community. So what's really critical is getting input from the community as to what features they think would be particularly restrictive or valuable or important to include here. Thank you.
So just to follow up briefly on what Peter said, so there's a lunchroom today where there's a meeting room where Peter will discuss this more and where you can provide that impact and feedback directly there. And also just to reiterate, this is an example of the sort of tool that interaction from the different Atlas communities with AWG. It's possible, I think this is going to be tremendously useful. I think we all know our students at postdoc spend an inordinate amount of time doing this sort of annotation work. And by working together in other areas as well, we can help to develop tools that are really going to benefit the whole community. So I hope that this stimulates you in each of your different tissue atlas groups to come and engage with us. We're not totally scary computational people, at least only Dana. And she is very scary. No, thank you very much.
Thank you.